This is GK. Welcome to King's Bio Production Academy. Let's understand about the RNA structure. What's RNA in full? This is an abbreviation, RNA, and that's why I've written in capital letters. So it's what? Ribonucleic acid. I'm saying RNA is what? Ribonucleic acid. Ribonucleic acid. That is on its full, ribonucleic acid. That is RNA structure. What is the RNA structure made up of? The RNA, the components of RNA. The components of what? RNA. Components of the RNA. Of which the components? RNA is made up of structures called nucleotides. It will be composed of what? Structure of chain called what? So we are saying RNA is made up of nucleotides. These are the components that makes up what? An RNA. So we are saying it's made up of nucleotides. So which are these nucleotides? The nuclear what? The nucleotides. The nucleotides, we have 3 nucleotides. Which are these nucleotides? We have the ribose sugar. We have a phosphate group. And the last one, we have the nitrogenous base. Nitrogenous bases. We are saying we have a nucleotide structure, and my diagram here is my nucleotide structure. You can check, we have a circular structure. You have a circular what? Structure. You have something of is it a pentagon or what do we call it? I'm thinking about this one. Then we have the nucleotides. So we have the phosphate group, the pentose sugar, that is the ribose, not pentose but ribose sugar. So we are saying this one, the far end is the phosphate molecule. Then we have this structure here, that is our ribose sugar. Then these ones are nitrogenous bases. We have 4 nitrogenous bases. So we are saying these ones are what? Nitrogenous bases, but they are attached to one at a go. That is one. I've attached this one. I can draw another one. Let me attach to that the first one. This one. I can draw, let me draw the 4th. Then I connect with this second one. Then the last one, let me draw this one, then I draw with a square. So this one, we are saying this one is the phosphate group molecule, this one is the ribose, and this one is, let me make the first one to be adenine, I make the second one to be guanine, I make this one to be cytosine, and I make this one to be uracil. I'll get to explain it. Those are nitrogenous bases. So we are saying the nitrogenous, so it's about a phosphate molecule with a ribose sugar getting attached to one of the what? Of these are the nitrogenous bases, and therefore, and the 4 nitrogenous bases. Now that is what it will take us to the next point to understand about these nitrogenous bases. Which are the nitrogenous bases of RNA? Nitrogenous bases of RNA. And our nitrogenous bases of RNA include 1. Adenine, abbreviated by capital A. 2. we have a guanine, abbreviated by G. Then we have not thiamine, but we have uracil, abbreviated by U, capital. Then the fourth, we have cytosine, abbreviated by capital C. Those are the four nitrogenous bases in these structure that we're speaking. And you can check what we have done here. So this one is guanine, cytosine, and uracil. In terms of their attachment or pairing, A bears with uracil. Then guanine bears with cytosine. We are saying they are in pairs, and the pairing, cytosine can only bear with guanine. The pair of uracil will be adenine. In RNA, we speak about uracil. In DNA, we speak about thiamine. So the thiamine, instead of thiamine, in RNA, you replace thiamine with uracil. That is it. Then we have now the types of RNA. We have types of what? Types of RNA. And we have number one, messenger RNA. From the term messenger, the one that carries message. So we are saying it's messenger RNA. Their work is to carry the information from the DNA then to the cytoplasm, not to the cytoplasm, but to the ribosomes in the cytoplasm, so that those proteins which are being carried can be synthesized. That is the messenger RNA. Carried the information from the DNA to which point? From the DNA to the ribosomes for the synthesis. Then we have the second one, and the second one is the transfer RNA. Transfer RNA. And this transfers RNA, we are saying here, is RNA. And our abbreviation for that is T small, then RNA. And the transfer is just to carry the information. This messenger, it was to convey the information. Transferring is from this point to the next point, and you can check where we are transferring from. It's from the DNA to the cytoplasm, then to the ribosome. That will be done by the transfer RNA. This is just a messenger, then this one is a conveyor. Then we have the last one, which is the ribosomal RNA. Ribosomal RNA. And it's small r, capital RNA, capital. That is the types of RNA. That is the types of what? RNA. That's the types of RNA. And this one is for the synthesis. After the coding, this that we are speaking, guanine, cytosine, and uracil, they're in coded form, and they're amino acids. And these amino acids, they will be in 3 pairs, and in 3 pairs they'll form a God. So what we are coding. What we do apply in the computer system about codes, it was, I think, that knowledge was gained from DNA and the RNA forms. That is it. So we are speaking about now. Let's go to the last point, which are the functions of RNA. Functions of RNA. But again, there is something that I forgot here. This one is a single strand. RNA is a single strand. And we're saying we'll be having the nucleotides along this lining. And we are saying, let's say, adenine, guanine, uracil, adenine, cytosine, uracil, adenine. Those are a single strand that we are speaking about. The single what? Strand that we are speaking about. Then the last one I'm saying, let's discuss about functions of RNA. Functions of RNA. Which are the functions of RNA? We just have 2 functions of RNA. The transfer of the genetic information. Transfer of genetic information. That is the first role of RNA, and the second one is protein synthesis. When we speak about protein synthesis, we are saying we have these what have we said? We have this we had the 3 types of chromosome of the RNA. That was messenger, transfer, and the ribosomal. The ribosomal role was to do what? To synthesize the proteins, and these proteins are synthesized to what? Amino acids. And which are those amino acids? Those nitrogenous bases. That's what we are speaking about. And the RNA is the one that will do that. The transfer of the genetic information. How is it being transferred? We have said from the messenger RNA via the transfer RNA, then to the ribosomal RNA, where now we will do this protein synthesis. That is the functions of that RNA. Then now, in terms of the pairing, we will form, you understand, we have the nucleotide. This is a one nucleotide structure. If I want to make now a polynucleotide, I'll just feed this one. This is guanine. I'll have to feed with the cytosine. So this one is the fitting of the cytosine. Then I draw this one. Then hope you understand it. So this is a pairing. And what am I pairing? Cytosine with guanine. And when I pair this cytosine with the guanine, I form something called what? Polynucleotide. So polynucleotide is just a long chain of many nucleotides. Thank you for your time. You can subscribe so that you can learn more and get engaged. My next slide will be on the DNA. Thank you very much.